Most people today have the misconception that the only DNA present within the human body is encased within the nucleus. However, this is incorrect. Supporting the theory of the mitochondria originally being its own separate organism, it also contains its own DNA within its lipid membranes. One key difference of the mitochondrial DNA is that the replication occurs at a constant rate independent of the cell cycle. This process is known as relaxed replication. It maintains a constant number of copies of mitochondrial DNA at all times. Another key difference of the mitochondrial DNA is that it is small and circular in shape, most similar in structure to what is seen within plasmids, and that they lack the presence of any introns. Within this small circular double-stranded structure, there is only one major non-coding region which contains most of the sequence elements that promote its replication and transcription. The replication process within mitochondrial DNA will always start at the ORI-H origin. To begin this process, there is a region upstream of the origin site known as LSP that, when transcribed, generates the primers necessary to begin replication. These RNA products form what is known as R loops, where the RNase primers stably bind to the template DNA and displace the non-template strand. These R loops anchor to what is known as a conserved sequence box, otherwise known as CSB, which is present between the LSP region and the ORI-H origin. Once anchored, the R loops are then processed by a nuclease known as RNA SEH1 to generate a 3' RNA end that polymerase gamma can then use to anchor and start to synthesize the new DNA of the H strand. As the replication fork begins to form, mitochondrial single-stranded DNA binding protein attaches to protect the separated DNA strands from reforming and also assists in the attraction of the mitochondria's version of helicase known as twinkle, which will unwind the remaining connected DNA in the 5' to 3' direction. Twinkle is assisted by topoisomerase IA, otherwise known as TOP3A, to prevent supercoiling of the DNA in the process of the H-strand replication. There are two regions in which the H-strand is known for replication to terminate. One region is known as the TAS region, and the other is termination around CSB2, which is within the original CSB where the R loop was anchored, leading to a full replication of the entire H strand of the DNA. For the replication of the L strand, starting from the origin point, ORI-L, the replication of the H strand must first pass this point. This is a key process as the displacement of the lagging strand allows for a stem loop structure to form, which exposes a region known as poly T-stretch in single-stranded form to attach POL-RMT, which is another form of polymerase that not only assists in creating the primers, but also blocks MTSSB from binding to the initiation site of the L strand replication. From there, replication occurs similarly as to the H strand. The main difference is that twinkle is not used for the replication of the L strand, nor is topoisomerase used, which leads to a supercoiling of the structure. To terminate the replication process, A nucleus that we saw earlier in the formation of the R loops, RNA SEH1, will come in to remove the remaining of the RNA primers, along with being assisted by a DNA-specific nuclease known as MGME1 that will remove the small tails left by polymerase gamma to create ligatable regions. These regions will be sealed by a ligase known as LIG3, creating a supercoiled structure with 2 complete sets of mitochondrial DNA. The separation of the two, along with the removal of the supercoiled structure, is done by a topoisomerase that we saw earlier, known as TOP3, which will deconcatenate the 2 structures and seal the nicks made, creating 2 separate complete sets of mitochondrial DNA and also the completion of the replication process.